Hello friends, welcome back to my YouTube channel Nestin Parasale. So today I'll be discussing the answers for those questions that has been asked for this Icmr JRF examination. The first question is that which one of the following is most likely to be found in the reticular endothelial cell? It is histoplasma capsulatum, and this histoplasma capsulatum is responsible for causing an eye disease called as histoplasmosis, and it is a dimorphic fungi which is acting as a parasitic organism, and it will be parasite this particular reticuloendothelial cells. Okay, if you want in-depth analysis, you can refer this particular article, friends. Okay. So the next question is with regarding to X and V factors required by which genus of bacteria. It is required by this Haemophilus genus, friends, and this particular Haemophilus influenza generally require two growth factor. One is called as X factor and the name is porphyrin, and another factor is called as V factor, which is SNAT. Okay, generally X and V is a bacterial test which is employed in microbiology field in order to determining the Haemophilus influenza differentiation. Okay, the next question is autoinfection is seen in life cycle of which parasite? It is seen in the life cycle of this Tiania solium, which is a pork tapeworm. Okay, so autoinfection is seen in the life cycle of this particular parasite. Okay. Next question is sterilization on 3 successive days by steaming at 100 degrees Celsius for 30 minutes is called as. Okay, so what does mean by that particular sterilization technique? It is called as tyndalization. Okay, so these things we had studied from 6th standard itself. So it is also called as fractional sterilization or germination inactivation or induced germination. It is an alternative sterilization method, and in this process we will be heating a particular substances for multiple time, and in between each time there will be a resting period, and it was given by Tyndall scientist in the year 1877. Next, mesophils are the bacteria that grow within a temperature range of, though these mesophils, they have a capacity to grow between the temperature of 15 degrees Celsius to 45 degrees Celsius. So the correct answer is 20 to 45 degrees Celsius. The extent of which a person's behavior, taking medicine, following a diet, executing life changes correspond with recommendation from a healthcare provider. So this process is called as adherence. So these things you don't need to have any particular knowledge. If you have general knowledge itself, you can able to answer this question. A tablet have no active ingredient and does not have any pharmacological action, but still it is used to improve the patient condition. It is called as placebo. So this particular tablet is generally used for a clinical trial. It is an inert substance which have no capacity to react with any kind of thing, and they are used for controls. In a clinical trial, they will be used in a control. At the same time, they are also used to improve the patient condition. So how this placebo will be improving person condition means they will be reducing anxiety. Okay, so if a person is taking this placebo and he will be expecting to feel better, this will be reducing a stress level. That is, stress causing chemical like adrenaline concentration will be reduced. At the same time, brain chemical, this placebos will be triggering the release of body's natural own pain reliever. So this is how this placebo drug will have a peculiar role in improving a person condition. Okay, next, your friend who is otherwise healthy or planning to travel to mountain to height above 8,000 feet above sea level. Which of the following medications would you recommend to carry with him? Ultimately, the correct medication is acetazolamide. So if you refer the CDC website, you can be able to see acetazolamide is generally taken for a person who is going in this particular mountain region and all. And the mechanism itself, the drug work by, they will be acidifying the blood. At the same time, they will be reducing this respiratory alkalosis. Okay, so this is how the drug is being generally used. Okay. Next question is with regarding to an high octanol water partial coefficient is required to maintain slightly to make the drug more permeable to blood-brain barrier. Of course, all the drug cannot able to enter into blood, cannot able to cross this BBB layer, because there is an epithelial tight junction will be present between this blood capillary endothelium. So that's why the drugs cannot able to enter into this BBB. But few drugs which is based on this high octanol and water partial, you can think about a person who intaking alcohol. And if you think about that particular concept, you can be able to answer correct for this question. Okay, so because alcohol has a capacity to cause this blood-brain barrier. Next, which are the following anti-diabetic drugs can cause vitamin B12 deficiency? The correct answer is metformin. Metformin is a drug which is suggested for type 2 diabetic patient, and the recent research was conducted in one particular university where they just find out the person who are taking this particular metformin is an anti-diabetic drug, there is a vitamin B12 deficiency, and for those persons who are deficient in this particular vitamin D, vitamin B12, there is high capacity for him to nerve damage and brain disorders. Okay. The volume of air inspired and expired during normal expiration is called as tidal volume. Next, which of the following statement is false about RBC? So, they are biconcave, of course, correct. They are non-nucleated, correct, and they are formed in red bone marrow, correct. But here they had given they have a lifespan of 60 days, but RBC the lifespan is 120 days. We have studied this in our 8th standard itself. So, what is the thing is that if you are going to prepare for next year, refer NCERT book from 6th to 12th standard. That's well sufficient for you. A single molecule of hemoglobin can transport up to it is 4 molecule of oxygen they can able to transport. The hormone involved rhythmic activities such as day, night and seasonal changes, it is ultimately melatonin. Okay, because these particular hormones, cyclin is a cell cycle hormone or cell cycle protein, they are involved in cell cycle checkpoint and all. Estrogen and calcitonin, they don't have any a role in involved in controlling rhythmic activities and also. The correct answer is melatonin. Okay. Next, kinoplasm is made up of copy of kinoplasm is made up of a mitochondrial DNA. Single step large mutation leading to speciation is called as saltation, because this saltation is completely an opposite concept of Charles Darwin. Charles Darwin says that evolution generally occurs in a gradual manner, but this particular saltation, which is called a single step large mutation, which was given by a scientist called Hugo de Veris. Okay, so this process is called a single step large mutation called as saltation. Okay. The last time itself we discussed when we answered key. So, the total amount of adenine time in the double stranded DNA is 45%. So, 80 is 45, remaining is 100 minus 45, it is 55%. Here they are asking only the amount of guanine. So, if you divide the 55 by 2, you will be getting 27.5. Next, how many duplexes are obtained from one duplex DNA after 10 cycle of PCR? It is simple formula, 2 to the power of n is number of cycles. So, the answer is 1024. A single nucleotide substitution, that is C has been replaced by A, has taken place in a gene, but the protein sequence is not altered. What is the type of variant? Okay, so you can refer this particular website which has given us synonymous mutation, which is really giving the synonymous variant. Is a change in DNA sequence that code for amino acid in a protein sequence, but they does not change the encoded amino acid. This is due to a property called as redundancy of a genetic code. What does mean by this particular redundancy? That is, multiple multiple codons will code for a same amino acid. Like if you take the codon for glycine, GGT, GGA, GGC, GGG, all will be coding for glycine. So if there is any synonymous mutation occurring, then they cannot able to change the protein coding sequences. So here that's a correct answer is synonymous variant. Okay, because a single nucleotide substitution has been taken place in a gene, but the protein sequence is not altered. Okay. Next, which of the wrongly pair? It is ultimately nucleic acid. The bond present is phosphodiester bond. Okay, so here they had given hydrogen bond, whereas glycosidic bond is present between polysaccharide, peptide bond is present between protein, and phosphate linkage is present between phospholipids. Okay. Next, what does this HGVS stands for? So it is Human Genome Variation Society. So here you can able to see this particular society is present in Australia. Okay, so it is present in Australia. Next, which of the what type of amplification takes place in sequencing by synthesis technique? It is bridge amplification. So, the best example of the sequencing by synthesis technique is that it is an Illumina sequencing technique. So, if you check on the Illumina sequencing procedure, first they will be preparing a genomic DNA, next they will be attaching a DNA to the Illumina cell. This is called as Illumina flow cell. The next process is bridge amplification, where when you add an unlabeled nucleotide and the enzyme, they will be initiating a solid phase bridge amplification. So, this is the bridge amplification, where you can able to see the structure of bridge. Next, the core of spliceosome complex is formed by, the core is formed by snRNA. It is not SNO RNA. SN means small nuclear RNA. It is present in spliceosome complex, whereas if you given small nuclear RNA, it is wrong option, because SNO is different from SN. DNA RNA polymerase differ in all except. So, they have a error rate. Next, the nucleotide substrate they incorporate, of course. DNA have a separate nucleotide, they will be incorporating. RNA will be incorporating ribonucleotide, and the processivity is also different. DNA polymerase will have a high processivity when you compare with the RNA polymerase, and the same DNA and RNA polymerase they will be they won't differ in the type of chemical reaction catalyst. The type of chemical reaction the same. In that what I mean to say is that they will be catalyzing a phosphodiester bond formation. Okay, so that's a type of chemical reaction they will be catalyzed. So both are anyway they are going to catalyze a chemical reaction called as phosphodiester bond formation. On a ribosome, the mRNA is read from. So it is 5 prime to 3 prime, that is from N terminal to C terminal. So if you check on the mechanism of translation means you are able to see from 5 prime to 3 prime the direction will be going. So this is how the mRNA will be read and protein will be synthesized. So the next question is with regarding to which of the following that does not encompasses reverse genetics. So what does mean by reverse genetics? So you should know forward genetics and reverse genetic. Forward genetic means they will be identifying a mutation and they will be linking a mutation to an observed disease. What is mean by reverse genetics? In this reverse genetic process, they will be inducing a mutation in a particular model or in any organism. Okay, and they will be studying a role of a particular disease. If you check genome editing, creating a gene knockout mice, introducing mutation, all are example or all are methods of this reverse genetic process. Whereas novel gene discovery by genetic screen is not a reverse genetic process, it is a forward genetic process. A smallest unit of genetic material which upon mutation produces a phenotypic effect is called as muton. Which of the following conditions of peroxisomal disorder? It is Zellweger syndrome. Which of the following repair pathway can faithfully repair a double stranded break? It is homologous recombination, which is a repair pathway that is going to repair this DS break, that is double stranded break. Okay. Next thing is regarding if the father in a family has a disease while a mother is normal, the disease is not only inherited to daughter and not to son. The disease name is the sex-linked dominant. Okay. So in that it cannot be autosomal dominant or autosomal recessive, friends, because if the disease has to be autosomal dominant, then it should be equally inherited to daughters and sons. But in this particular thing they had given, it is inherited only to daughters and not to sons. Okay, so the thing is that it is a sex links. Okay, so now you need to know whether sex linked recessive or sex linked dominant. The sex linked dominant is the correct option. So during this dominant inheritance, the abnormal gene, so it will be occurring when one abnormal gene from one parent can cause a disease, even though the matching gene. So here the daughters, they have two X chromosomes. So one they will be getting from mother, that will be a normal. Another X chromosome definitely they will be getting from a father. And since father is having a disease, so he will be transmitting the diseased X to all of the daughters. So it is sex-linked dominant. Mendel does not observe linkage due to his third law called as law of independent assortment. Next, who coined the term linkage? It was coined by T.H. Morgan. Actually, Bateson and Punnett, they first they first tell about this concept of linkage, but the term was given by this Morgan only. Next, an antimicrobial biocontrol agent that is used to control butterfly caterpillars is Bacillus thuringiensis. Okay, so it is generally used as a biocontrol agent to control the butterfly pest. A fall in glomerular filtration rate, they generally activate juxtagomerular cells, macula densa and lacy cells, because these JX cell will be releasing a renin enzyme. Okay, so when there is a fall in GFR, then they will be activating all the cell to release a hormone called renin. Next, which of the following vertebrae is present in is formed from 4 vertebrae? It is ultimately coccyx. Okay, so it is a tailbone which is a triangular structure and it is present in the bottom of spine. It is made up of a fusion of 4 vertebrae. Next, sarcomere is so sarcomere is a contractile unit of muscle and it is a repeated structure present in a muscle cell. Okay, generally actin and myosin, it will be present in this particular sarcomere only. So it is a segment which is present between 2 Z line. We had studied this from our 11th standard zoology itself. Okay. End of long bones are covered with cartilages. Next, the joint between carpal bones are called as gliding joints, whereas the joint between knee joints are called as hinge joint. Between this, knee bone, it is called as hinge joints. Okay. Next, how many fragments will be generated on digestion of closed circular DNA with a restriction enzyme? They are giving closed circular DNA with a restriction enzyme having 6 recognition site. The answer is 6. In case of linear DNA, so how many, there is a question that how many fragments will be generated on digestion of a linear DNA with a restriction enzyme having 6 recognition site. For linear DNA, it will be generating 5 fragments. Since it's a closed circular DNA, it will be generating a 6 fragment. Next, so this is again a question to challenge. If you don't know how many questions I had challenge means, you can watch my last video. Last video just make a detailed analysis. 3 important questions that a student should challenge. Okay. Next thing, biotechnologies consisting the use of biological systems like bacteria for manufacturing, transformation or degradation of molecule through enzymatic or fermentation process for industrial purposes is called as it is white biotechnology, whereas yellow biotechnology, it is a biotechnology with insects, blue biotechnology is a marine biotechnology and green biotechnology is a agriculture biotechnology. Which of the following is not a component of downstream processing? It is ultimately expression. These things you no need to study about downstream processing and all. If you have general knowledge, then you can able to easily answer those questions. So separation, preservation, purification are a component of this downstream processing. Plasmids are used as a cloning vector for which of the following reasons? Because plasmids are extra chromosomal circular DNA. They have a capacity to autonomously replicate. So they have an immense capacity to self-replicate inside the bacterial cell. The first clinical application of gene therapy over 4-year-old girl was done on adenosine deaminase deficiency. So do you know the name of the 4-year-old girl? The name is Ashanti De Silvia. Actually, she is still living. Okay, so she is still living an active life today. Okay, the experiment was done on National Institute of Health on the year 1990. So if the question has been asked for some other exams, it will be really helpful for you. The diagram which shows the arrangement of metaphasic chromosome according to the position of centromere is called as ideogram, because these ideograms are generally useful for locating a position of individual genes on chromosome. Okay. How many type of histones are found in nature? It is 5 types. That is H1, H2A, H2B, H3, H4. During a DNA this is again a question that you should concentrate and you should challenge this question. Why I am saying means during DNA replication, the synthesis of lagging strand of DNA results in a fragment called as Okazaki fragment. But here they had given leading strand. So this is the wrong option. Okay. The next question is a role of aminopterin in hacked medium cells. So aminopterin is generally used in order to block the de novo pathway. Okay. So guanidinium group is present in arginine amino acid. Okay. Which component of protein absorbs UV light at 280 nanometer? It is indole group of tryptophan. Okay. So here they had given imidazole group of tryptophan, but imidazole group is not present in tryptophan, it is present in histidine. And also this histidine, that is imidazole group of histidine and indole group of tryptophan has a capacity to absorb at 280 nanometer, and the sulfur group of cysteine has a capacity to absorb at 260 nanometer. Sodium fluoride is used in sugar estimation. Inhibit which of the enzyme? So generally this sodium fluoride is act in order to inhibit a glycolysis pathway, and generally this particular chemical has an action of preventing or inhibiting an enolase enzyme. So enolase is an important enzyme that is catalyzing the final step of glycolysis. And if you ask me why we need to stop glycolysis, ultimately so it is used for sugar estimation. Only if we stop the glycolysis temporarily, only we can able to know the exact or actual estimation of glucose concentration. Which of the following is required for porphyrin synthesis? Okay, so in order to synthesize porphyrin, two things are needed. One is glycine and another one is succinate. So both are acting as a precursor of delta aminolevulinic acid. So after this delta aminolevulinic acid, they will be converted into protoporphyrin and then porphyrin. So this glycine is acting as an important precursor. In tricarboxylic acetic acid, fumarase is converted into malase. So fumarase is converted into malase by an enzyme called as fumarase. So for that particular enzyme action, it needs water. Pyridoxal phosphate is used as a coenzyme in a various biological reaction. So this pyridoxal phosphate is generally used in a reaction called as transamination, and is used to transfer the amino group from an alpha amino acid to a ketocarboxylic acid. Next. Yearly ambulation is in a post-operative period prevents which complexes? So yearly ambulation is nothing but after a surgery or after the operation, the person will be asking to get out from a bed, and they will be asking them to do few light action, like they will be asking you to stand or walk for a while. It is generally used to prevent this deep vein thrombosis, which is nothing but blood clot in deep veins. Okay. So the next 1-year-old is suspected for bacterial meningitis. So, for which lumbar puncture was performed? Which of the following laboratory finding of spinal fluid would support the diagnosis? So, they will be during this particular lumbar puncture, they will be injecting they will be taking a spinal tap, okay, where they will be taking few samples from spinal cord. Okay, and during the bacterial meningitis, which is caused by bacteria, okay friends. So when there is an infection in membrane called meninges is an important layer which protects the spinal cord and brain. So that cases it will be having a bacterial meningitis, and during this particular disease condition, in the spinal cord fluid there will be high protein concentration. So that's why they will be taking, because during this particular condition there will be breakage of your blood brain barrier, so there will be an increased amount of immunoglobulins will be secreted. So in order to deter detect the level of immunoglobulins, which is otherwise a protein. They will be determining the elevated protein level. Growth chart or road to health is a visible display of weight for age. Which of the following RNA constitute 90% of total RNA? It is your ribosomal RNA, which is make up 90%. So, in the sequence alignment by BLAST, each word from query sequence is typically, so the word size for protein sequence is 3 and a word size for nucleotide sequences is 11. If the purpose is to calculate the probability of one event and a second event, the odds score for the event or it should be multiplied. The cryopreservants used for T cells consist of, it is 10% DMSO in fetal bovine serum. DMSO means dimethyl sulfoxide. It has a capacity to membrane penetrating activity. So, they have a capacity to water displacement property. So, they are generally added along with the media in order to reduce the ice crystal formation, thereby preventing the cell from cell death. Okay. Next, COVID-19 is caused by positive. So, COVID-19 is caused by coronavirus, which is having a positive sense single stranded RNA. Okay. So, Yamanaka factors are nothing but they are SOX2, KLF2, OCT3,4 and C-MIX. They are used for this induced pluripotent cell reprogramming. So, these are 4 transcription factors that have a capacity to convert a somatic cells from a terminally differentiated state into an embryonic cell. So, don't forget these transcription factors, they are called as Yamanaka factor. Hybridomas are immortalized for production of unlimited supply of monoclonal antibodies. So, how they will be producing monoclonal antibodies? There will be a antigen producing B cells and there will be a myeloma cell. So, they will be fusing both the cells. So, generally electrofusions or PEG, that is polyethylene glycol will be used. Okay. So, cell fusion is a technique. Niemann-Pick disease is a lipid storage disease. It's a rare and inherited disorder and it affects the body ability to metabolize your cholesterol and lipid. This disease is also an example of lysosomal storage disease. The electrophoresis technique, which is used to separate large fragment of DNA, is called as pulse field gel electrophoresis. Polyacrylamide gel electrophoresis is used for separation of proteins. So, pulse field is generally used. So, the entire genetic material, that is entire genomic DNA, we can able to separate by using this pulse field electrophoresis, where the DNA will be first digested with a restriction enzyme, then they will be running the gel matrix under electric field. So, they will be changing the direction of electric field. Okay. The process by which normal serum enhances the phagocytosis is called as opsonization. So, opsonization is again an innate immune response. So, the process is handled by a protein called opsonin. So, they will be tagging a foreign pathogen for eliminating. Okay, elimination by phagocytosis. So, without opsonin, there will be a repulsion between a cell wall and a phagocytosis. So, that's why this opsonization is very important to enhance this phagocytosis process. They will be breaking the repelling force existing between the cell wall of pathogen and phagocytosis. Okay, cluster of differentiation. What is mean by that? It is present on the surface of cell. Okay, so they are the protein that are present on the surface of cell. Actually, this particular CD is a nomenclature system which is used for identifying and classifying the antigen that are found on the cell wall of leukocytes. A ligand for TLR. TLR means toll-like receptor. They are the class of protein that play an important role in innate immune response. So, this receptor have a single pass membrane spanning receptor. They will be recognized as structure from microorganisms. So, the ligand for this TLR3 is double stranded RNA. Okay. So, which of the following mechanism allows a complete absorption of glucose from intestinal lumen into cytosol of enteric? It is the sodium glucose co-transporter 1. Okay. This particular transporter is also involved in reabsorption of glucose also. Okay. So, this is the easiest thing. They had given a molecular mass of IgG, it is 150 kDa, and the antigen and antigen complex in the presence of antigen excess is 300 Dalton. So, the formula is that to find the they are asking to find the molecular mass of antigen. The molecular mass of antigen is equal to molecular mass of antigen and antibody minus molecular mass of antibody. That is 300 minus 150. So, you will be getting 150 kilodalton. But you know, one antibody it have two binding site, two site to bind. So, you need to divide 150 by 2. So, the correct option is 75 kilodalton. All type of chemical forces are involved in antigen-antibody interaction except covalent bond, because the covalent bond is the strongest bond, whereas van der Waals forces, hydrogen bond and hydrophobic bond are generally present between antigen and antibody interaction, because these bonds are non-covalent at the same time they are reversible. Okay. Next. Predominant antibody present in serum, it is IgG. The second predominant antibody is IgA. Which of the following method cannot be used to determine the three-dimensional structure? So, X-ray, NMR, cryo-electron microscopy we can use for determining three-dimensional structure. But Ramachandran plot, it is used as two-dimensional plot which is used for determining the torsion angles of protein. During peak summer and winter, the frog takes shelter into deep burrows to protect them from extreme heat and cold. This is called as, the summer sleep is called as estivation and the winter sleep is called as hibernation. The same question is also asked in CSR examination, friends. Okay, so which of the following pairs of organism are called as eurycotylic? So eurycotylic organism means the organism which will be excreting the nitrogenous wastes in the form of uric acid. An example include birds, insects and lizards. Okay. So ureotylic means the organism which will be excreting urea, that is example is fishes. The factor that lead to founders effect in population is called as genetic drift. Okay, so founders effect and population bottleneck are the two examples of genetic drift. So what is mean by genetic drift? It's a random process which will be leading to a large change in the population, okay, within a short period of time. So germinal centers of the lymph node and spleen perform which of the functions? So germinal centers are present in lymph node and spleen. They are the site of antigenic stimulation of mature B cells. So already just mentioned you, lymph node and spleen are secondary lymph node organ in which the activation of B cells will be there, and in this germinal center the antigen activated B cells will diversify their immunoglobulin gene and they will be generating a high affinity antibodies. Okay, the type of cleavage found in mammals is rotational cleavage. E. coli is used as an indicator organism to determine, it is used to determine the water pollution as well as sewage pollution, and also only if the water is polluted with fecal matter, because E. coli is highly present in fecus. Certain bacteria live in soil, convert nitrate to nitrate and then into free nitrogen. So such bacteria are termed as, so such bacteria are called as denitrifying bacteria, friends. So example includes Pseudomonas aeruginosa, micrococcus denitrification. There is another bacterium called as nitrogen fixing bacteria which will be fixing the nitrogen from the atmosphere to the plant. It will be helping the plant. Okay. Next, the prebiotic atmosphere of the earth was a reducing nature. It was transformed into oxidizing atmosphere of present day due to emergence of, it was due to emergence of cyanobacteria which is called as blue-green bacteria. So this cyanobacteria, they had evolved before 3 million years ago and they perform oxygenic photosynthesis, which means they will be using an water to get hydrogen and oxygen. Fossils are generally found in, sedimentary rocks only. They are used, lichens are used as an atmospheric pollutant only, which means this lichen are pollution susceptible, so they will be growing only in a non-pollutant area. They won't grow in an area that was polluted. Kranz anatomy is present in leaves of C4 plant. So which one of the following is an example of homology? It is ultimately thrones of this bougainvillea and tendrils of cucurbita. So homologous organs means they will be having a different function and the structure will be different, but they have a common origin. If you take both the plant, they are the modified from a branch, or they are both are modified branches which are present in the axilla of leaf. Okay, so thorns, they have a different function to protect the leaf from invaders, and tendrils have a different function, like they are used for climbing. Okay, so they originate from a divergent structure. Whereas if you take this eye of octopus and mammals, they are both are used for vision, but both are having a different origin. And same if you take flippers in penguins and dolphin, penguin and dolphin, they have a similar function, which means they are analogous organ, but they have a different origin. Okay, so the correct option is option number 4. Hormones which is responsible for root micropropagation, it is auxin, where cytokines is used for shoot propagation, and abscisic acid is used for stress responses, and this gibberellin are used for germination, senescence and flower development. So long plant are able to stand due to the presence of this sclerenchyma, because this sclerenchyma will be providing a mechanical support as well as strength to plant due to the presence of lignin compound. Okay, so this sclerenchyma is generally present in non-growing organs of the plant like bark. Which of the following reproduce sexually once in a lifetime? Ultimately the answer is banana plant. So you can also challenge this question also, friends. Which of the following is a breed of cattle? It is Ayrshire, it is a breed of cattle. It is a Scotland cattle breed, and you can, the other quality of the breed is high, and this particular breed you can find with a red and white marking. Whereas this Gagas is a breed of chicken, and this Kadakanath is also a breed of chicken. Whereas this Campti is a breed of lobster. Which of the following is a marine fish? It is Hilsa. Okay, Hilsa is a marine fish. So generally you can find it is an export quality fish, and it is a costly fish where you can find them in a Bengal areas. Male reproductive tract developed from Wolffian duck. So Wolffian duck, the other name is mesonephric duck. Okay, they will be giving rise to male reproductive structure like vasa differentia, semina vesicle. Okay, whereas this Mullerian duck, other name is paramesonephric duck. It will be giving a female reproductive structure like fallopian tubule and uterus. Which of the following glycoprotein layer is surrounding the oocyte? It is zona pellucida. Zona pellucida is a glycoprotein layer, and it is responsible for inducing the sperm to undergo acrosomal reaction, and next role is that this particular layer will be preventing the sperm from fertilizing your eggs. Okay, So next, sebaceous gland secrete to which mode of secretion? It is hollow grain secretion. So the hollow grain secretion means in this type of secretion, the entire cell will be breaking down to release a sebum. Okay, and sebaceous gland is the best example of hollow grain secretions. Thank you friends. Thanks for watching this video.